Welcome, Samantha Hughes. Very welcome to our live stream here from iXperium Health to Health Valley event. You are a researcher and a teacher at HAN Biocentre, and you will tell us something in the next half hour about health benefits of functional foods. I'm very curious about that. Thank you for being here, and it's all yours.
Thank you very much. Thank you for having me. Yes, I'm going to give you a bit of a talk about some of the things we're doing in the research lab here in the HAN Biocentre at the moment. And first I have to introduce to you the model organism that we work on. So obviously humans are the best model for which we want to explore how foods or drugs might affect us. But there are reasons why we can't actually study in humans, and there are a number of different model organisms which we can actually work with. So mice or cell culture, zebrafish and flies, for example. But my organism of choice is the 1 millimeter long nematode, c-elegance. So hopefully I'm clicking or not. There we go. So humans and worms are very similar. You might not think it, but they are. We both have a skin, so a thick barrier protecting us from the outside world. We have intestines, a brain, nervous system, exactly the same as the worms do as well. And this is one of the reasons why working with nematodes is actually really useful for us. There are a number of other things. Again, my pointer is broken. There we go. So unfortunately the video doesn't work right now, but what you'd be seeing here would be a little worm wiggling across the screen. So as I said, these are 1 millimeter long. They have a very short hermaphroditic life cycle. So they self-fertilize. We can have 250 genetically identical offspring from a single adult worm. And what this means is we can very quickly do large-scale studies. That's a bit of the boring stuff, but also their systems biology is highly comparable to us. They also have conserved molecular pathways to us, so between worms and humans. I'll come back to that later. And the genetics are very identical. So in terms of our DNA, we have 80% homology between us and a worm, and that's pretty high. So what that means is we're able to do a lot of investigations at the genetic level to see what might happen in the worms and relate that to us as humans. And of course, there are a huge array of molecular tools that we can use to analyze different things in the worms. We can do CRISPR-Cas9, single gene knockdowns, we can do optogenetics where you take out particular genes due to light, lots of things. But what I'm going to tell you about today, no, I'm still not working. Click please. Yes. What I'm going to tell you about today is just one of the many topics that I research in my lab here in the HAN Biocentre. And I'm going to talk to you about two of our main projects. One is looking at the health benefits of functional foods called Tart cherry juice, and another is our work that we've been working with some collaborators in the Radboud UMC and at Liverpool John Moores Hospital on green tea and the health benefits of green tea. So, I'm working again. So why do we research into the health effects of functional foods? Well, functional foods are very important to us, we need them for life, but they also provide various different benefits to us. They might help us develop better, have better movement, so for example, better recovery after exercise. Ideally, we want to eat lots of fruit and vegetables that help us live longer and healthier lives, which is the main focus of my research. But also we want to try and find foods which might help us lower our risk of being obese or getting diabetes. And we also look for now for a lot of foodstuffs that are beneficial in terms of neuroprevention, so we can reduce our chances of getting Alzheimer's or Parkinson's disease, and also for cancer research. So what foods might help us have a better chance of surviving a cancer diagnosis. So there's a whole array of things that we are looking at, but I'm going to, like I say, going to tell you about two of the particular projects we work on. So first I have to, I'm sorry, remind you that aging is inevitable. We all age. It even happens to these 1 millimeter long nematodes. And what you're looking at here is a graph of lifespan. At 0 time point, this is a young worm just before sexual maturity, and it's quite a happy, healthy worm. It takes 3 days to reach this L4 stage, after which it will spend 3 days laying its 250 viable offspring, and then the worm hits menopause and it will just live out its life for the next 3 weeks, or 2 to 3 weeks. And as it lives out its life, it will just show similar signs of aging to us. So its movement will slow down, it will stop eating so much, and it even gets wrinkly, right? So because we know this happens, and this is very robust, this line that you're seeing on the graph, this is actually the average or the actual population decrease in age over about 250, 300 individual animals. And to test how we can check if a worm is alive or dead is, imagine you have this 1 millimeter long worm on a Petri dish on a plate under a microscope, you tap it on the head. And if it responds to that tap by wiggling its head back, then you know it's alive. And if it doesn't respond, it's dead. So you count the death. What we're able to do, because we have this very known, consistent lifespan graph, is we're able to look for the effects of foods that shift this graph left or right. So can we increase lifespan? And one of the things I'm going to tell you about today is this cherry juice, specifically Tart cherry juice. And we wanted to know if these cherries actually did extend life. Unfortunately, we can't feed the worms cherries as whole pieces because you can't just dot them on the plates. So we actually supplement the plates with an active concentrate, so cherry active concentrate. So you have to imagine we have this plate, we have plates with low levels of tart cherry juice and high levels of tart cherry juice. And this cherry juice is the same type of dosing as you would expect in a normal human. So it's equating to low, medium and high dosage of humans drinking the cherry juice. What was really strange to us, I think, here we go, is we saw some striking differences. If you took a little bit of cherry juice, shown here in the blue line, you see that the graph has shifted to the right. So you actually get an extension to lifespan. So that's brilliant. However, if you take a lot of cherry juice, so every day you drink a lot of cherry juice, you're actually not doing so well and you actually have a shortened lifespan. So this was really strange and we weren't quite sure why this was the case. So we decided to investigate this into more detail. And what we found, quite interestingly, was that tart cherry juice alters fat content. Here what you're looking at are two pictures of worms which have their intestines stained with a Nile red dye, which binds fat droplets within the animal's intestines. And you can see at the top is the control worms, wild type worms, and then underneath is the worms with a lower, who have been exposed to the lower amount of cherry juice, but have a much higher level of tart cherry juice overall, a higher level of Nile red staining in their intestines. Right, so, and we have quantified this on the graph on the right. So this was really crazy to us, like why would these worms be more fat on something that's actually more healthy? We then looked at the fat content of worms which are had been exposed to even higher levels of cherry juice, and you see that the fat content goes back down again. Now this is really strange at first glance, and I'm going to say something slightly controversial here, but at least in worms, there is some evidence to show that those worms which live longer, genetically live longer, have actually got a higher level of fat. And it's all to do with energy balance and redox stasis. But I am not suggesting that you should eat lots and get fat just so you live longer, because that's not the connection it works, right? It's a bit more complicated. So from this, we were actually more interested in to see what specific type of fat is actually causing this effect. So which fat, are there a change in the fat content? And to do this, we connected with our chemists in the department and asked them to measure the fats in the worms, the triacylglyceride types. And here what you can see is the result, approximate result from a gas chromatogram. And we see that in the red squares, this is the fat content in the wild type worms that have not been exposed to the cherry juice. When we then exposed worms to cherry juice and measured their fat content, there is a very distinct change in the fat profiles. And what's particularly interesting is their 18.0, 18.1 and 18.3s. And why are these interesting? Well, these relate to the fats that are in a Mediterranean diet. So we know from literature and general talking around that if you have a diet which is a Mediterranean-based diet, then you have a different type of fat content, and these people are suspected to live longer. And so we're starting to tease out why this is the case and looking at the different fat contents. So that's one avenue of research that's still ongoing in my lab, but that's what we're looking at as well. Now, because because these worms are fatter, and because these worms are living longer, we started to think maybe there's something to do with a calorific restriction type effect happening as well. So what we wanted to do here was actually look to see, are the worms eating properly, and can we actually measure their calorie intake? And to do that, we basically look at the neuromuscular pump, which is the mouth and the throat, if you like, of the worm, the pharynx. Now normally you just count the number of times the worm chows down on the food, but in our lab we have a microfluidics device which allows us to generate basically mini worm ECGs. So what you're looking at here is the pumping frequency of the animal. So you take the worm, you put it into a little chamber with electrodes at either side of the animal, and then it automatically counts how often that worm is pumping. And from this data you can get things like pumping frequency and how many pumps there are per minute, and this gives you a lot of information about how healthy the worm is and if it's behaving normally. What was really surprising to us when we took our worms which were exposed to low levels of cherry juice is that the pump frequency went down, but the pump duration went up. And this is in comparison to wild type non-exposed worms. So what this suggests is that the worm is still eating, but it's taking fewer bites of food, if you like, and when it is biting food, it's taking longer to process that food. So the worm is actually undergoing a sort of calorie restriction diet, and we know that calorie restriction is also related to longevity and a longer lifespan. So together our data at the moment from the Tart cherry juice work shows that Tart cherry juice increases lifespan, and this is corresponding to an increase in fat content and a decrease in the neuromuscular function. And this is showing us that Tart cherry juice is acting as a calorie restriction mimic. And we now know how this acts, which metabolic pathways this is functioning through. This work has been done in collaboration with the University of Hertfordshire in the UK and is part of an ongoing work package that we're trying to really delve into the minutiae of what's actually happening and the mechanisms by which this is working. Okay, changing tack just a little bit. One of the great things that I find working at the HAN Biocentre is we're able to work at the interface in this so-called research triangle. What's this? This is basically allowing students to engage with, who are in education, to engage with researchers, active researchers, but also industry. So we basically get the students to do work that's relevant to a societal problem now, but also we allow them to explore this in a safe environment before they go out into the world of work. And I'm going to give you two different examples of where we've done this. The first is actually in our education. In the third year, there is a specialization called Plants for Food, Feed, Fiber, Fuel and the Future. And here the students actually obtain knowledge about the molecular biology of plants and modern breeding, and they do a little bit of CRISPR-Cas modification within the plants we have in the Biocentre. And they learn about how the secondary metabolites are produced by the plants, how they're used by us humans to treat diseases and improving our health, but even in production of biofuels. During the semester-long course, the students start presenting their research into how plants are used as secondary metabolites, as nutraceuticals, and students actually produce products such as posters and small student-led conferences to think about how they could best demonstrate their chosen crop to be more beneficial for society. And I've taken some screenshots of some posters that I think are particularly interesting, where students have come up with the idea of adding more spicy flavors to strawberries, or making coffee beans more healthy, so your daily quota of coffee becomes better for you than perhaps it currently is. And this is really exciting, and the students come up with some amazing ideas, which is really cool. But our teachers are actually looking for more industry collaborators. So if there's anyone out there who works on this or is interested in working with us to explore these aspects, then please get in touch, because we'd really love to hear from you. And another project we have, which is actually in my research lab with a student, and this is the connection with the Radboud and Liverpool John Moores, is we're actually looking at a student research project. So I had a student come to the lab to do a 6-month internship with me, and she was looking at the health benefits of green tea. So we know tea, green tea especially, is generally perceived to be very healthy, and we know that it has a lot of polyphenols, these antioxidants that makes it healthy. But tea is caffeine. There's caffeine in that tea, and caffeine might not be so healthy. So there's interest now into the decaffeinated green tea. So is this as healthy as regular tea, and can we measure the health benefits of this? And this work was done by an intern in my lab, Sarah, who you can see here. So she explored the fat content of the green tea. So she essentially brewed tea and fed it to the worms. And we were really happy to see that green tea does reduce fat content in all cases. So this is at low and high levels of green tea. So fantastic. Green tea, good. Take some green tea. We also found that it actually affects health span in a positive way. So again, my student looked at the lifespan extension compared to worms which had been exposed to tea and not. So the red line, you have control worms, no exposed. And in purple, you see worms which have been exposed to regular caffeinated green tea. And here there's no actual change in lifespan, not really. But, but if you, you can't quite see it, but if you expose worms to low levels of green tea, this decaffeinated green tea, there is an extension to lifespan, but it's very small. But there is one there, and it is significant, at least if you're a worm. And we're actually looking to see if this is also true in human studies as well. So that's part of our collaboration with Liverpool and with the Radboud. And because of this interaction between fat content and lifespan, we thought we should look at the insulin signaling pathway. You all, I hope, are aware that we need insulin in our lives to live healthier, and if there's a problem with the mechanisms, then you end up with diabetes. Worms have a highly conserved system here where we can actually specifically pinpoint different aspects of the insulin signaling pathway and look for defects. We explored 3 different parts of this pathway. So we looked at the change in gene expression, so how much genes were going up or down following exposure to the green tea. And what we see is that SOD1, which is a detoxification enzyme, which usually removes any free radicals. There's no real change in gene expression for this, as we expected. SKIN1, which is the nematode homologue of NERF2. This is in response to oxidative stress and has been shown to promote longevity. In worms which are exposed to decaffeinated green tea, they actually show an increase in the expression of SKIN1. And what we suspect is happening, that this is starting to signal pathways which are involved in longevity and starting to tell the worm, you know what, we can turn on our healthy pathways. And most interestingly, we actually see a huge, relatively huge, upregulation of DAF16. And DAF16 is the worm homologue of FOXO, the transcription factor which actually goes into the cells and activates genes which are involved in insulin signaling, longevity, and metabolism. So you see this massive upregulation of DAF16. And together this tells us that decaffeinated green tea is having a positive effect on various aspects of life and insulin signaling. At the moment, what we're doing is seeing if green tea provides an anti-diabetic effect. So we do have assays in the lab which we're still optimizing to try and test whether worms are able to survive high glucose levels. And if they can this actually be moderated by the exposure to green tea? So this is something we're exploring right now. We're also exploring whether we can actually measure the reactive oxygen species production in the worms before and after being exposed to green tea. Again, an ongoing project. So watch this space for the data that comes out from that. Therefore, in summary, we have shown some health benefits of green tea. We've shown that decaffeinated green tea at low and moderate levels does reduce fat and actually increase lifespan and provides some stress resistance, which is fantastic. And we're still progressing our research to look at how these pathways are being affected and what are the pathways that are being affected. overall, what I've hoped I've shown you today in the last 20 minutes is that c-elegance is a fantastic model system to explore the mechanisms that underpin health and disease. And I've only shown you a small snapshot of the work that we're doing in the Biocentre. We do a lot more. And we are able to integrate our topics into education and research at the HAN Biocentre, but we're always looking for industry collaborators, especially for the students to get more experience of the questions that industry have. And if you're interested, please get in touch, or even if you're interested in testing your compound. You know, we're always interested in exploring other foods and other metabolites that might have an effect. Okay, so with that, I'd like to thank you for listening, but I should also thank the people who've done the work. So of course our partners with the Green Tea Project, Dick Tyson's group at the Radboud and Katie Jane's group at Liverpool John Moores, and the Cherry Juice Project, which is still ongoing, which wouldn't be possible without Lindsay Bottoms. And of course all the lab members that I've been working with, but specifically Sarah, who has been integral to the Green Tea Project. So thank you very much, and I'll take any questions.
Thank you, Samantha. Very, very interesting. I know we work a long time already with c-elegance as a research worm, I can say it like that. Is it something we are specialized at HAN Biocentre, or is it used worldwide, this worm, for research?
Yeah, no, it's used worldwide. There are over 4,000 nematode labs across the world, so wherever you are, there will be a worm lab. And there's even a worm lab on the International Space Station right now as well. So we're even in space.
Oh, that's cool. And you're all connected? All c-elegance researchers are connected?
Yes, we're a very, very active community. We're very collaborative. We share strains. We ship them in the post to each other. We have a lot of meetings, topic meetings and group meetings with everyone. So yeah, really connected.
I think it's amazing that a 1 millimeter worm can be compared to us. We're complex beings, I always think, and we react, quite similar like the worm does. Yeah.
Well, what I think is also really exciting is the worms were at the forefront of so many technological advances. So the Human Genome Project, for example, we wanted to sequence the human genome, but that technology at the time was super ambitious, and they thought, well, what should we test to make this work? So they chose c-elegance. So we've actually are at the forefront of technology development. We were the first organism, apart from jellyfish, to get GFP in there, RNA interference knockdown, also first done in c-elegance. So it's not just research, it's technology development. You know, we're a really strong community for that as well.
Yeah, that's very cool. And what I wonder, because we are similar but also different, we also react different sometimes on food or drugs or whatever. Are the difference between worms? Do they also, I don't know so very much about these worms. Are there male and female worms? Are they different because we have different reaction on medicines before?
It's a good question, actually. So the worms are hermaphrodites, so they have both male and female parts. So, but in saying that, you can generate female worms and you can get male worms as well. And there are groups which do look at the differences between these populations. And there are also groups, there's a group at the Ver actually, who look at natural isolates. So the nematodes are not just lab-based, you can find them outside, under an apple tree, on the apples on the ground. So you can pick them in your garden if you like. So it's not that, it's not that they're so different to us. And people have shown different responses between them. But one of the nice things we can do, because the worms have so many babies basically, and they're all genetically identical, we're able to look at such large numbers that you see any population level changes, which is quite nice as well.
Yeah. And so they're also similar when you use c-elegance here in the Netherlands. They're similar to the ones which are in space now?
Yep, they're the same strain. We have the same strain, so they all come from the same
mother,
original. Yeah. Which are British actually. So yeah.
So how many, from my idea, how many strains are there worldwide? Millions or thousands or?
Probably. Yeah. I don't wish to put a number on that.
Okay.
We have a again, so it's showing how great our community is. If people make nematode strains, they deposit them in a stock centre, and they're available to anyone who wants them. So the stock centre is in the US. they're
then
funded,
you order.
and you order them.
Can
they
I
come
have
in
some
the
new
post.
worms, and then
Yeah.
you get a little box with 1 million worms.
Yeah, pretty much.
Ah,
Yeah.
yeah. I think it's very cool. Because I was also like it's getting wrinkles. It's getting slower, so that they really have in 3 weeks a whole lifetime.
Yep. The whole life, and
Yeah.
you see, so some of my research looks at Alzheimer's and Parkinson's disease, and you can really see them get the accumulations of the plaques, the same as humans do as humans age.
Also, then,
Yep.
and
We can do that.
there are little brains in the little worm.
Pretty
Oh.
much, yeah, kind of primitive, but yeah, so you can see them.
But
Yeah.
that's exciting to know that they also have plaques.
Yes. So
And
that's
so you can also research what food or drug can have for difference on the
Exactly.
metabolism
Yeah,
of the worm.
that's exactly what we're looking at.
Ah.
And also exercise, because we can exercise the animals, which sounds really strange. We give them a little mini treadmill and to work.
No,
on.
I was thinking, how do you tap a lot more often on the head?
No, it sounds very strange, but we can also do that. We can check memory and learning in c-elegance as well, so we can look at all these different aspects that are related to us.
But
in these
how
tiny
do
ways.
you exercise? And because I was still wondering, how do you tap a 1 millimeter worm on his head?
Yeah.
I would probably smash the worm. I'm sorry.
No.
But how do you exercise then?
So for tapping the worm on the head, we use a very thin platinum wire or an eyelash,
Okay.
and you literally tap them on the head. For the exercise, we put them in a liquid media, so they're like
okay, they have
moving
to
around.
swimming lessons.
Swimming lessons, and we can put them in through a gel, which is a bit like resistance training. And what has been shown by other groups, not by me, is that if you do this, the worms show the same changes as we would do if we were undergoing sport and exercise. So they change their fat metabolism, they change the use of glucose, they get tired and they can't do anything for a while after this.
You could make a whole television show about a worm.
That would be cool.
I mean, got lots of ideas. No, but it's amazing that you can make lifetimes of people in 3 weeks, and again with a lot of relatives, with a lot of comparison. Yes, it's really amazing.
I love
Well,
it.
thank you very much for this research. I know we will continue it.
Mhm.
You already did make the call to people. If you want to collaborate with us, just give a call.
Yep.
info@han.nl or Samantha Hughes. You can find you on our sites.
Yep.
And well, I think people can find us.
Yep.
Thank you very much. Really interesting.
Thank
topic.
you very
Thank
much
you.
for having me. Thank you for listening. Thank you.